Hello class. Today we will discuss another topic of colloids, that is purification of colloids. As in the earlier videos we have seen that the preparation of colloids and all. In preparation of colloids we are using many stabilizers such as electrolytes, peptizing agents and sucrose, glucose and all those things. So we have to remove all these stabilizers, or we can say the electrolytes which is present in the colloids. So for that purpose we are doing the purification of colloids to remove the excess amount of electrolytes which is present in the colloidal dispersion. Basically there are three methods by which we can do the purification of colloids. One is the dialysis method, second one is the electrodialysis method, and the third one is the ultrafiltration method. So we will study in detail one by one method. So first, coming to the dialysis method, this is the picture of dialysis method. You can see here from the dialysis method what we are doing, we are removing the ions, that is electrolytes, either the sodium or chloride ions, or the molecules, that is glucose or sucrose, which we are adding as a stabilizer present in that, through the diffusion process by using semi-permeable membrane. By using semi-permeable membrane, as the name indicates, semi-permeable, it permeates certain kind of molecules from that and it retains certain kind of molecules within it. So we are using semi-permeable membrane. The semi-permeable membrane may be the parchment paper, or we can say the gold bladder fish skin, or we can say fish bladder, or a gold beaten skin of large intestine of ox we can use. So this is the setup of dialysis method. What here we have to do, we have to take a test tube which is open on both the sides, not test tube, we have to use a tube on which the tube should be open on both the sides. One of the end of the tube is covered with the semi-permeable membrane, that is semi-permeable membrane, either the parchment paper or either the gold beaten fish bladder or gold beaten skin of large intestine of ox we can use as a semi-permeable membrane. So after doing this, what we have to do, we have to pour the colloidal dispersion. We have to pour the colloidal dispersion, for which we have to do the purification. Then later on, all this setup we have to put into another beaker or another large vessel in which there should be a continuous flow of water. You can see here I have shown water outlet and water inlet and water outlet. From here it is water inlet, and from here water will go out. So what happens, once we will keep this, or once we have poured the colloidal dispersion into this, the ions or the molecules which is present here in the colloidal dispersion will come out into the water, and that will be continuously flow out as we are continuously, this assembly is supplied with the water. And the solution which is present, in the colloidal dispersion, the solution which is called as a colloidal dispersion, here we are calling it as a diffusate. Diffusate means the solution which has to go through with the diffusion process, and the solution which come out, we will call it as a dialysate. Dialysate means the solution has been dialyzed and it has been come out. So this is about the dialysis process. The another second one, what I had told is electrodialysis. In electrodialysis, the basic principle is same as that of the dialysis. But the difference between the dialysis and electrodialysis, as in the name itself it says electrodialysis. Here we are applying the potential difference to speed up the process. In dialysis, it takes a time to dilysate or to purified the colloidal dispersion. So to speed up the process, what we are doing, we are having a potential difference, and this is the setup of electrodialysis. This is the setup. You can see same as that of the dialysis process, but the difference is here I have given the potential difference. You can see here, this is the both are the semi-permeable membrane which I have shown with the red marks. These are the semi-permeable membrane, and which I have shown with the blue pen. One will be the positive side and another will be the negative, that is the electrodes we have applied. Then once we are electrodes, that is cation to absorbs cation and anion. Then once we apply the potential difference, what happens? The ions that is electrolytes which is present in this will get purified by the electrodialysis process. But the advantage or the disadvantage of electrodialysis process is that only the ionic molecule can be purified, not the molecules. If you remember, at the beginning I have told purification means we are purifying ions as well as the molecules. Molecules means glucose, sucrose, which we are adding in the preparation of lyophobic colloids, especially in the lyophobic colloids, that can't be purified through the electrodialysis process. This is the main disadvantage you may get for 2 marks. Disadvantage of electrodialysis, or they will ask advantage of dialysis. In dialysis advantage, all kind of compounds such as ions, molecules can be purified, but whereas in electrodialysis only ionic molecules can be purified, not all the molecules. This is about the electrodialysis. The third one is the ultrafiltration. As the name indicates, ultrafiltration means here the setup is similar to that of the dialysis only, but the thing is, in spite of using semi-permeable membrane, here we are using ultrafiltrate paper. So what is ultrafiltrate paper means? If we are using filter paper, what happens? The pores which is present in the filter papers are very large, from which the colloidal solution will easily comes out due to the size. So what we are doing? We are reducing the size of the filter paper. How means? By taking colloidal solution, colloidal, not colloidal, colloid ion, ION last में आता है, colloidal solution. Then we are impregnating the filter paper. Then you may ask the question, what is impregnations? Impregnation is nothing but, say for example, this is the filter paper, this is the colloidal solution. I will dip the filter paper in this, then later on I will take out, and this filter paper is what I will do with, by keeping another 2 filter paper, I will press out, and so that later on what the filter paper I will get, this will be called it as a ultrafilter paper. From the ultrafilter paper, all the ions and molecules can be diffused out, but whatever the colloidal solution will be there, it will remains as such as a slimy, we will see. So how the setup will be? The setup will be like this. This is the ultrafiltration setup. You can see here I have added colloidal solution. This is the ultrafilter paper. Ultrafilter paper. All the solutions which contains ions, molecules or glucose will be comes out here, and so you can see I have written medium colloidal dispersion plus electrolytes. Here colloidal dispersion, colloidal impregnation. The colloidal solution which is to be dialysate will comes out and will remains here colloidal dispersion as a slimy. This colloidal dispersion we will take out, then we will mix up with a suitable dispersion medium and prepare the colloidal dispersion. This is about the ultrafiltration, and the application of purification is, as I had told, for the purification we are using, and to determine the binding of protein with the, or to study the protein kinetics of protein binding. Protein binding we will be studying, and the most important this one application is hemodialysis. Hemodialysis is the important application. Hope you have understood 3 methods: dialysis, electrodialysis and ultrafiltration in the purification. This you can expect for 5 or 2 marks. Thank you.